So this lecture is primarily going to be for the Transfection Lab. Now, with the school being closed, we essentially are not going to be able to perform anything with the transfection, but I want to go through the lecture that I would normally have given you in class so that you understand the theory behind the background and essentially what we're going to do, and I'm going to write this up so that you have a written concept of what I want from this lab write-up, is I'm going to go through the theory behind the transfection and what we're hoping with the cell differentiation. So it's kind of going to be like 2 lectures back to back. I normally would give one week, and then the following week I'd give the second half, but I'm essentially going to do both lectures at the same time. And then what I'm going to have you do is write up a lab report, and I'm going to give you some data that was done from previous classes, and I'm going to have you analyze that data and basically do a simple write-up to explain what you understood from it. We're going to do the same concept of intro and materials and methods and results and then discussion. So the only unfortunate part that you won't get is actually doing the transfection. However, transfections are pretty simple. They're not too difficult, especially the one we were going to do. So understanding the theory behind it, the protocol itself is something that takes maybe half an hour to do, and we can definitely look at having you get that experience later on when we come back to the school. But for now, we're just going to have you walk you through the theory of that. So let me kind of walk you through ultimately what transfections are. It's basically introducing DNA into the organism, but unlike the transformation with bacteria, it's a little more complex with eukaryotic cells. And the reason for that is because we can't really do a heat shock method. It kills the eukaryotic cells, so we have to use other methods to be able to transfect them with that. Now, the first thing that we need to do is, what vector are we going to use? And the vector that we use actually has a couple of different factors to it. Obviously, it has that ampicillin-resistant gene, usually the beta-lactamase, that, if I can get my thing to work here. The beta-lactamase gene that allows you to transform it into bacteria and then grow it up. And then, of course, you've got some other elements in here as well. We've got the GFP element that you normally see to be able to be as a marker. And then we've also got another gene that we've inserted into here. And so with this plasmid, we've actually introduced a gene called Plin2. And I'm going to explain later on what that gene ultimately does, but it's a transcription factor for now. So what we want to do in this first part of the lab is we want to say, how can we introduce this plasmid into eukaryotic cells? Because you've already done this with prokaryotic cells, with bacteria, and that's easy to do. But the big question then becomes, how do we get them into eukaryotic cells? Well, there's a couple of different methods. You can actually do what we call a physical injection, where you actually have this needle and you put it into the cell. Now, this is typically done with embryos, where they are able to do somatic cell nuclear transfer. They're able to use CRISPR or other things to physically introduce this into the cells. And that takes some finesse because you ultimately have to have all the right equipment to be able to do this on a microscopic level. It's really difficult to do, but is one of the more tried and true methods, especially when you're dealing with embryonic manipulation. But that's not always the easiest thing to do, especially when you're dealing with cell culture. Another thing you can do is adenovirus, or basically a retrovirus, where you can infect the cells with a virus that will incorporate that DNA into the cells. Now, you can't really do that with a plasmid. Generally, you have to have, if you do create an adenovirus, you start with the plasmid, you have to use a different method to transfect the cells, and then that can create the virus that can then do the rest. And we used to have this, but we ran out of it and we weren't able to reestablish the titer, which is the number of viral particles. It really wasn't a difficult lab to do because you would just put the liquid in with the media and it would infect the cells. So it was very, very easy. So if you have the ability to construct a virus and actually infect the cells, it's very simple to transfect the cells with your DNA, but to get there takes a while. So that's not really one that's used quite often because it's very specific on what you're doing. Now, there's other methods like electroporation, And electroporation is basically you put the plasmid next to the cells and you run a current through the cells. Very, very quick current, couple of milliseconds. You usually have to have a machine that does this. I did this when I was doing my my PhD work, where we had this machine that did 5 microsecond bursts, and we were able to have this little machine that was able to put just tiny, tiny amounts of DNA. You really don't need a lot when you're doing this process. Now, this really only works well if you have a large grouping of cells, especially when you're dealing with embryos and you have like this, three-dimensional embryo. That's when this becomes really useful. But when you're dealing with adherent cells or even suspension cells, it's a little more difficult to get the DNA to sit on one side of the cell and actually pass that electricity through them. Because what happens when you electrocute the cells, it's very much like the heat shock method for the bacteria. It temporarily allows the cells to kind of have these little cracks. It pushes the DNA into the cell, and then it closes up. So you can't have too high of a voltage or too much of it, for too long. Otherwise, it just fries the cells. And I've done that before. And so you got to have very specific parameters for electroporation to work. I definitely used it a lot, but in this class for cell culture, it's hit and miss. It's very difficult to kind of do. So other methods, you can actually sonicate the cells. I've never done that before, but sonication can temporarily kind of permeate the cells and allow for that to go through. I do know of another colleague that used to do that and had some measure of success with it. So you use sound vibrations to kind of, crack open the cell and let the DNA go in. But that also can kill the cells pretty easily if you don't do it right. And then one of the more tried and true methods that is almost always used now is what we call Lipo Fection. Now, this is kind of the way to go, because a Lipo Fection is basically you take your plasmid and you incubate it with this reagent that kind of forms this micelle, right, that surrounds the DNA. So this is like a cell membrane micelle. So it's got the hydrophobic region and the hydrophilic region. And when you incubate your DNA with this lipofectamine reagent, we usually use what we call turbofectin. But when you incubate those together, they'll actually form these small lipid spheres around the DNA. And then when you incubate these with the cells, here's the cell membrane here that is also phospholipid bilayer, then it actually just kind of allows this to just enter right in, almost through endocytosis, where it'll recognize this and it will just pull it in and bring that into the cell, and then the cell will actually start transcribing and using the information. So this is actually a very, very good method, very simple to do. All you have to do is essentially combine the plasmid with the reagent, let them sit for a couple of minutes. Now, one of the keys to making this process work is that you have to use what we call serum-free medium. Now, think about that for a second. What does it mean to be serum-free? Well, that means that you don't use FBS. And the reason for that is because albumin, which is essentially that, if I can spell right, that protein that's found in fetal bovine serum that is found in your blood as well, typically will grab fats and things of that sort and help in some of the transport. It's not like the HDL and the LDL molecules that you have, but these proteins will grab drugs, they'll grab fats, they'll grab various things. Well, remember that this albumin also deactivated your trypsin, EDTA, that you were using to trypsinize the cells, and it got rid of the enzyme. Well, the same problem occurs here. If you have media that has the FBS, it will actually prevent this membrane from forming around it, because the lipofectin reagent and the albumin will interact with one another, thus neutralizing your ability to make these micelles. So that's typically why when we do a transfection, it says serum-free. You got to make sure that it doesn't have any FBS in it, otherwise it just won't work. The other thing that you have to do when doing this process is you have to make sure that you don't have a lot of liquid. And the reason for that is the following. Let me go ahead and erase most of this. So you've got your plate and you've got your cells sitting at the bottom here. And if you have a lot of liquid up here and you put your micelles, your transformed agents in there, they're just going to be kind of free floating around here. Some of them will kind of settle down, but they don't usually do that. And so the reason why you want to have such a low amount of liquid when you're doing a transfection is that ensures that these lipids, these micelles, really don't have anywhere to go. And so they're going to be sitting just right on top of the cells. Now this lends itself for drying out. So this is why usually a good rocker in the incubator to make sure that this is just kind of staying right on top of the cells. Otherwise the middle tends to get dry, the cells die, and you really only start to see transformations along the edge. So it's one of those things that you got to be particular about doing it. Normally the transfection lasts minimum 4 hours to 24 hours. Obviously, this is kind of you got to do it at least that long for it to work, but up to that long. The problem with going this long for 24 hours is it can dry out, it can cause problems and whatnot. See, it depends whether you have a rocker or not in doing that process. So those are kind of the variables that I would want you to talk about when you're doing your write-up in some of the problems that can exist with the transformation protocol. So that's why when you're doing the transformation, number one, you don't use the serum media, you use serum-free media, so that when you combine your DNA with this reagent, we call it turbofectin, when you combine those two together, they'll essentially form that combined micelle that's necessary to be able to go right into the cells. And this is why we have a low amount of solution. We do it for a certain amount of time period. The other thing about this is the cells have to be about 50 to 70% confluent. And the reason for that is they have to be actively dividing through mitosis. If you don't, if you're about 90 to 100% and they're pretty much done, you're not going to get a good transformation with these micelles because they really only allow the cells in when they go through this process of mitosis to be able to allow them to kind of merge with the membrane and take in that plasmid. And so this was one of the things we were going to do was we were going to plate the cells on Monday and shoot for 50% confluency on Wednesday. You'd come in Wednesday, you'd basically do the transfection protocol, which was to put the plasmid in the media, let it sit for a little bit, put the turbofectin in with there, let it sit a little bit, let them combine, and then take that, add it to your cells. You first pull out all the media, and then you just add this small amount of media to your cells. Typically, we would do these in 96 well plates because that ensures that with these really, really small wells, that even though you have a little bit of media, they're not prone to drying out. And so that's why we like to do it in these little 96 well plates. It all depends upon what you're trying to do. We're not trying to get a huge amount of cells transfected. We just wanted to make sure that it worked and that you're going to be able to get some observable cells. And again, you can still get hundreds of thousands of cells in these 96 well plates. So it's not something, generally if you use a larger plate like the ones we did our cell culture with, you would get, a lot of cells transfected, but the big question is why? What are you looking for? We're not looking to transfect all the cells. The whole purpose of this lab was primarily to introduce that plasmid that had that gene PLIN2. Now, let's talk about that plasmid and whatnot. For our transfection protocol, we would use a 96 well plate and we would cause some of them to be transfected with this plasmid with PLIN2. And then, of course, we'd have a couple of controls. Some would just have the GFP control and others would have the PLIN2 vector. And we would split it up that way and you'd be able to compare the two. So you'd have some cells that were transfected with the plasmid that had GFP, PLIN2, and the BLA gene. And then this one was the same plasmid, it just was missing the PLIN2. So this was our controlled GFP. So they both were going to glow, but the difference was what happens when these cells take on this plasmid, and they start expressing this transcription factor PLIN2. So the first part of this lab was the transfection. Basically, we would have done the transfection on Wednesday, and we would have just let them grow till the following week. The following week, I would have basically given you a lecture, as I'm about to do now, on differentiation. And then we were going to make observations on those cells, and I'm going to give you data on Canvas under Files showing you what the cells would have looked like at that point. So let's talk about differentiation. Those that have already taken my developmental biology class already know all of this, but I'm just going to kind of walk you through the basics. So when you start off with a single cell, the sperm and the egg fertilized, and you get that zygote is what we call undifferentiated. It has no fate. It can become any cell, both embryonic and extra-embryonic, and it essentially goes through the rounds of mitosis. And after several days, you end up having kind of two groups of cells. You get this outer group of cells, which we call the trophoblast, and you have this inner group of cells, which we call the inner cell mass. These outer cells, they take on the fate of becoming the extra-embryonic tissue, the placenta, the amniotic sac, the umbilical cord. But these cells, the inner cell mass, these cells ultimately, if I can get my pen working here, right? These cells will become brain, blood, hair, liver, kidneys, skin, all of that. These are all of the cells of your body. And so these are what we call the embryonic stem cells. These are the cells that when you have, stem cell research with, these are the embryonic cells. And so these cells still have the full potential to become every cell in the body. They're not completely what we call totipotent. Totipotent is basically the potential to become any type of cell, and that's really the zygote. When you get into these cells, then they become what we call pluripotent. Pluripotent, these are the embryonic stem cells. They can become pretty much any cell in the body. But as they go through this process and eventually start forming the skin and the liver and the tissues, then they eventually become what we call multipotent. Now, these are what we call the adult stem cells. These are the cells that, like our bone marrow, that regenerate your red blood cells, white blood cells, and platelets. Or they're the basal cells of your skin that regenerate the top layer of your skin or whatnot. Those are the adult stem cells. Those are the ones that basically keep you alive throughout your entire life. Now, these have a limited potential. Skin can't normally become anything but skin. But because they're similar to other cells like neurons, you can actually tweak these stem cells just a little bit and actually turn them into neurons. They've actually been very successful with various gene transformations where they're able to cause skin stem cells to become neural stem cells. And that's been a fascinating aspect of research. And that was the whole reason behind doing this transfection, is to show you academic research with stem cells and cell culture and what you can learn from all of that. Now, what we were going to do is we were essentially going to take kidney cells. These were the fibroblast cells that are used to repair kidneys. We were going to introduce a transcription factor called PLIN2. This is a gene that will actually turn on a host of other genes that are dormant but still available in the cell. And we were going to turn them into fat cells, or at least start making them behave like fat cells, turn them into adipocytes. And so adipose tissue is essentially the tissue that will store triglycerides, and they do that by essentially creating these lipid nodules, these areas that can store the fat in these environments. And so the difference is when you look at your kidney cells or the fibroblast cells, here's the nucleus and all of that, and the rest of that. When you look at these and you see them, with the GFP in them, you really will just kind of see the whole cell glowing. And that's what we'll expect with the control cells, where you essentially transfect them with the plasmid. It's just going to create GFP. It's going to glow. It's basically going to create it in the cytoplasm, and you're going to see most of the cell glowing. However, in the cells that have the PLIN2 gene active, not only are they going to glow, but they're also going to turn on additional genes that have been dormant but still available that will cause the cell to start bringing in and turning the sugars into fats and storing those fats in these lipid dropules. And so what you'll see in the pictures is you'll see these GFP protein, but then you'll see these huge gaps or areas where there's nothing, and they look like just kind of blank spaces, but in reality, those are the lipid nodules that you would typically see in fat cells. So this has always been an amazing experiment to show how you take cells from this state and you can actually cause them to change their role, their fate. And this fundamentally comes down to which genes are active. And that's really the difference between all of these different cells. They all come from the same origin, but fundamentally they have different genes turned on. Now those genes sometimes are so dormant that you can't even turn them on because of chromatin remodeling. And so if the genes are inaccessible, even if you were to introduce this transcription factor, you wouldn't be getting this process. But because these two tissues come from about the same source, then they're very similar to one another, very much like how skin and neurons actually are very similar to one another. And thus you can turn on some of the genes in the skin cells and turn them into neurons. Excuse my chicken scratch here. But when you get cells that are very similar and closely related to one another in terms of what genes have been turned on and off, then it allows you to actually change some of their fate by if you could put the right gene in there. And this has been shown to be very effective in giving an example of differentiation of those. And so that's ultimately what we were going to be showing with this. The transfection was the cell culture procedure, very simple, very easy. And the rest of it was just waiting for the cells to turn on that gene and for these to show the difference between the kidney cells that became adipocytes versus the control ones that are going to glow but really not have any morphological changes. That's one of the ways which you can tell the difference between cells and their cell type is the morphology of them. And so these lipid dropules are a substantial difference, and you're going to see those in some of the pictures that I post. So the big question is, how do I want you to write this up? Obviously, I've given you a lot of theory, a lot of background. So let's look at what I'm looking at for the write-up here. Unfortunately, like I said, we can't do this lab. We may be able to in a future semester. If you guys haven't graduated yet, we'll have you come back and maybe do this. Colleen obviously does some transfection in her lab with the plasmid, and they look at something a little different than ours. But fundamentally, it's the same procedure with the turbofectin, and so it's not too different. But obviously, for our purposes, we're looking at the end result that's a little different. So intro in your lab write-up. I want you to talk about transfection. Specifically, I want you to look at explain the different methods that can be used for transfection. I went over those: electroporation, physical injection, and virus, retroviral infection. And then, of course, spend most of your time on Lipo Fection. Just give a little background ultimately on what Lipo Fection is and how you're able to introduce the plasmid. I also want you to talk about differentiation, right? So, and this is a lab that there's going to be two parts to the intro. First part is transfection. Second part is the theory behind what differentiation is. So, talk about how stem cells are undifferentiated. They haven't taken on that fate. They have the potential to. Just depends upon what genes get turned on during that time. And then, of course, talk about how adult stem cells can be reprogrammed. And so, obviously, the fibroblast cells aren't an adult stem cell line, but it follows the same procedure that when you put some genetics in and you can turn certain things on using transcription factors, that we can actually get the cell to respond differently because it's now creating a different protein profile. In this case, we're getting kidney cells to look and behave like fat cells, like adipocytes. Okay. As far as the materials and methods section, basically describe the fundamentals of the transfection protocol. So let's go over those again real quick. Basically, you culture your cells so they're about 50 to 70% confluent. That's the basic standard. And when you come in, as long as they're within that range, you can do the transfection protocol. You basically take a small amount of DNA. So make sure you know that you've got the control GFP and you've got the plin2. And really the only difference between these two vectors is this gene is added on top of this. So they're virtually the same. The only difference is this gene is in the plin2 gene, and other than that, they're identical to one another. And so these are the two plasmids we use for the transfection. Obviously, the way we set it up is we're going to have a 96-well plate. We're going to have some controls. So a couple of wells, we would have done the GFP control, and the other wells, we would have done the plin2. We would have basically cultured the plasmids with turbofectin. Okay, so that's the reagent that we use. You just use like a microliter of this with a couple microliters of the plasmid. You mix them together, let them sit for a little bit to form those micelles, and then you put them in there. Obviously, running time is up to 24 hours, but minimum 4 hours. So that's kind of the basic material method procedure that you would have on this write-up. Results, these essentially are going to be the pictures. So make sure you look under Files, under Canvas, and I'm going to have a couple of pictures, and one will say Control GFP, and it'll show a cell that's just kind of glowing green. And the other one will say plin2, and this will actually show that cell with those lipid dropules in it with the GFP in other areas and whatnot. They're very distinct from one another. So obviously you'll label them control, and then this is the plin2 plasmid. And then under discussion for your write-up, basically talk about some of the things that could go wrong, especially when you're dealing with transfection, things like the serum-free medium. That's something that people mess up on all the time when doing this process in terms of efficiency of that. Also, in your discussion, look at the differences and just explain some of the concepts that we talked about with differentiation. Obviously, why do cells look different? This is because different genes are turned on. Remember, plin2 is a transcription factor. So it's not the plin2 gene that's actually making these lipid dropules in this. It's turning on other genes that are then creating these dropules, and because there's many genes involved in this process of storing fat, creating fat, and ultimately changing the morphology of that. Now, you don't need to know which genes exactly are being turned on, but definitely talk a bit about transcription factors. These, again, are the genes, especially in the early stages of cellular differentiation, that control which genes are turned on and which ones aren't. If those transcription factors are there, they're going to create a cascade of different genes being turned on and other things being turned off. And that's ultimately how cells will take on that particular fate of becoming a stem cell, or going from a stem cell to a terminal, skin cell or neuron or whatever the case may be. So as you talk about the discussion, go through some of the issues that are pretty prevalent in the transfection protocol. Talk about how things can dry out and how you would, solve that with maybe a rocker and whatnot. And then obviously when you get into some of the theory behind that. So it's not too difficult of a write-up. Again, it's in one of the more amazing labs that unfortunately we didn't get to do, but you will be able to see the end results and kind of do that mock write-up for me. And then I will grade that for you as you get to the end of that. And again, if you have any questions, just let me know, and I'll kind of help you walk you through it for this mock-up lab.